Today we'll talk about the law of segregation. This law of segregation is also called as law of purity of gametes. What this law of segregation is? Law of segregation states that at the time of gamete formation, that is at the time of gametogenesis, the two alleles of a gene separate from each other and each gamete receives only one allele of the gene. So for example, we have this capital T and small t. These are two alleles of a gene. At the time of gamete formation, the two alleles of a gene separate and each gamete receives only one allele of the gene. So this is law of segregation. Law of segregation states that what is the law of segregation? Law of segregation states that at the time of gamete formation, at the time of gamete formation, gametogenesis, the two alleles of a gene separate from each other. They segregate from each other. They separate from each other. They segregate from each other. And each gamete receives only one allele out of an allelic pair. So out of two alleles, the gametes receive only one allele. So this is known as law of segregation. That means when gametes are to be formed, the alleles of a gene separate from each other and gametes receive only one allele. And this is known as law of segregation. This law of segregation is also known as law of purity of gametes, Why it is known as law of purity of gametes? Why it is called as law of purity of gametes? Because gametes are always pure. Gametes are always pure. Gametes have only one allele out of two alleles. Only one allele out of two alleles. So gametes are always pure. So this represents gametes are pure. That is, out of two alleles, gametes have only one allele. Gametes receive only one allele out of two alleles. They are always pure. That is why it is known as law of purity of gametes. Law of segregation is also known as law of purity of gametes. This law of segregation, it is considered as the Mendel's first law of inheritance. It is Mendel's first law of inheritance. It is Mendel's first law of inheritance. And this law of segregation, it is explained by means of a monohybrid cross. So we'll explain it with the help of a monohybrid cross, this law of segregation or law of purity of gametes. So we have parental generation, tall pea plant, it is crossed with dwarf pea plant. So we have genotype, capital T, small t, small t. So this is genotype. Obviously this is phenotype. Then gametes. So which law will get applicable? Law of segregation. Law of segregation. That means the two alleles of a gene separate from each other at the time of gamete formation, and gamete receives only one allele out of two alleles. This is known as law of segregation. So this is one allele, this is one allele. That means in a gamete, out of two alleles, only one allele is present. So we say that gametes are always pure. That is why law of segregation is also known as law of purity of gametes. This gamete has only one allele out of two alleles. Gamete receives only one allele out of two alleles. So gametes are always pure. So this is known as law of segregation, law of purity of gametes. Same is the case here. Gamete formation, law of segregation will get applied. So then we have offsprings, what are called as F1 generation. So capital T, small t. Same is the case here, capital T, small t. So obviously this is tall pea plant. This is tall pea plant. Why tall character has appeared? It is law of dominance. Only one, according to law of dominance, the character which appears in F1 generation, that is a dominant character. So tall character, tallness is a dominant trait over the dwarf character, but this is heterozygous. That is, had two dissimilar alleles for the trait. Then we'll go for the selfing. When individuals of the same phenotype, of the same generation, they are crossed with each other, of the same colony, they are crossed with each other. So we have got capital T, or we can write down here F1 crossed with F1. So we'll get capital T, small t. This pea plant will be crossed with this pea plant. Tall pea plant will be crossed with tall pea plant. And these are gametes. Gamete, only one allele out of two alleles in a gamete. Only one allele out of two alleles in a gamete. Only one allele out of two alleles in a gamete. So this is gamete formation. Again, law of segregation will apply. So then we have Punnett square to get the F2 generation. Have male gametes here, female gametes this side. We have capital T, small t. This is capital T, small t, capital T, capital T, capital T, small t, capital T, small t, small t, small t. This. Now we'll take here how many tall is to dwarf. So tall we have 1, 2 and 3. 3 tall, 3 and 1 dwarf. So this is tall, this is tall, this is tall, and this is dwarf. So this is phenotypic ratio. Now genotypic ratio. We have genotypic ratio capital T 1, then capital T small t. We have 3 genotypes, 1, 2, these are similar, that is why we will take only 1, and small t. So we have how many individuals are these? There are 1. How many these capital T and small t genotype individuals? 1 and 2. These are 2:1. So genotypic ratio is 1:2:1, and phenotypic ratio is 3:1. And this is how is law of segregation explained. This law of segregation, it is universally acceptable law. This law of segregation is out of law of dominance, law of segregation, law of independent assortment. This law of segregation, it is universally applicable. That means it is applicable to plants as well as animals. However, this law, it is applicable to all diploid organisms which form haploid gametes by means of meiosis to reproduce sexually. But this law of segregation is unapplicable to haploids, unapplicable to haploids like bacteria. They are haploids. They have only 1 allele out of 2 alleles. That is why this law of segregation is applicable to all the diploid organisms, the plants and animals, because these diploid organisms at the time of gamete formation, at the time of formation of gametes, they form haploid gametes by means of meiosis to reproduce sexually. And this law of segregation is unapplicable to haploids. It does not apply to organisms which are haploids, for example, bacteria. Thanks.